go into our class today. If you're latecomers, we will do the present afterwards. What we discussed in the last class about the muscle tissue, just we finished earlier, but according to our syllabus, we have to cover a little bit of the muscle tissue again. So that's what I'm going to do today, and then we will review for your exam. So at least this exam, I expect everybody should get an A grade. Okay. Let us go for the document camera, and then I'll explain to you. Okay. Did we discuss the last class about muscle tissue? See, normally we used to say, I don't know which saying that I heard this of muscles of iron and nerves of steel. Have you ever heard of that one? Muscles of iron and nerves of steel. Have you ever heard of that one? Why do you say it like that? Muscles of iron and nerves of steel. What the iron and steel will do? Steel will give the strength, right? That's more, and muscles will help to move one place to the other, the arm. So here, if you do they used to say, I think in a gymnastics, everything, so you have to have the muscles more flexible and it should be very strong and everything. Now, how strong it is and how it works, so the mechanism behind it. So that's what we are going to see today, of a little bit of mechanism of muscle. So we discuss about the muscle tissue in a different types. Number one, we know about the skeletal muscle. Number two, did with the smooth muscle. Number three, cardiac. Of course, you know all the tissue, skeletal muscle tissue, smooth muscle tissue, and cardiac muscle tissue. And we also have seen under the microscope and also the staining procedure, the muscle tissue. Yesterday, some of you have done the work with color. What color have you seen in the muscle staining? Mason trichrome stain. You observe some red color, right? So that's for the muscle tissue. That's a light microscope. Okay. We'll go into some some detail of individual muscle cells, and then we will study. Generally, if I say a cell, you know about nucleus and the membrane, this membrane, plasma membrane, and this nucleus, and this is cytoplasm. And then you have mitochondria, and then Golgi and all the. This is the general cells. But in a muscle cells, instead of cytoplasm, we used to have, like this is being converted, this will called a sarcoplasm. Sarcoplasm. One word, sarcoplasm. Okay. And I'm just a single muscle cell. This I'm explaining to you. This nucleus, the one which will just moving in one side. Okay, nucleus will go one side. But the rest of the area, which is occupied only with cytoplasm, but here the sarcoplasm. I'll draw in a different way now. Like okay, just in a cartoon. So I just put that there's a membrane, and here I put a nucleus, the blue one, nucleus. Okay, because single cell, but nucleus is pushing on one side. Okay, and then the cytoplasm here we call it as a sarcoplasm. I just draw this is called a muscle cell. For the time being, ignore about the either skeletal muscle or cardiac muscle or smooth muscle. Just ignore that. Just muscle cell. So nucleus is one side, and the membrane, plasma membrane or plasma lemma, this one in the muscle cell we call the a sarcomere or sarcolemma. Sarcolemma. Membrane. S-A-R-C-O, sarcolemma. L-E-M-A, lemma. And then the cytoplasm, as I mentioned before, sarcoplasm. Sarcoplasm. Okay. So the main function of muscle cell, which I told you earlier, what is the function of muscle? It involves in the contraction and relaxation, right? The function is contraction. Contract and relax. That's the function generally. So the function really, each cell will have this characteristic. Do you understand that? Each cell should have the characteristic of contract and relax. This is the function. On this cell, what part or which one is responsible for this contraction and relaxation? In the cell, we know the cytoplasm, and there's a proteins which are responsible for that. And these proteins are present in the sarcoplasm here. So I just draw like the red dot, okay? They are the protein. So these proteins are responsible for contracting the cell and relaxing the cell, okay? You have other stuff like mitochondria is there, and Golgi is also there. But they are arranging in a one a pack of all these protein. So these proteins, in other words, we call it as an actin and myosins. Actin, one protein, and myosin, the other protein. So you have to recollect. A cell which has the actin myosin as a protein which is present in sarcoplasm or in the cytoplasm of the muscle cell, and it pushed the nucleus one side. So you have a major area is occupied by the sarcoplasm, and the actin and myosin are the proteins. And this protein, how this protein is arranged? This protein is arranged in a very good fashion. In a sense, when this cell needs to contract and relax, the protein which is present inside that do the function. You follow? Now I'm going to see how this protein is being arranged in the in a muscle cell, and then we will see a little bit of its function. Because once you understand the architecture of this actin and myosin, and then it is easy to comprehend, understand the skeletal muscle or smooth muscle or cardiac muscle. So, just I want to go on to the next the details of this actin and myosin. You just bear with me. You have to draw whatever I am drawing on here, so that will help you. But also I have given in your figures, so that's also good. You can go through it. But I'm going to draw the pattern, and then it's very important to understand, because I'm going to ask some questions for you, short answer questions too. Okay. Just I'll draw and then I'll explain to you. Okay. So this is one. Okay. I'm writing the arrangement of the actin and myosin. Okay. So I'm just the arrangement of these fibers here, these dots, you know. So how inside the cell is arranged by electron microscopically. So if you see the dark one, which is which I call it as a thick one. Okay, here. This one. Just I want to put a really thick one. Otherwise, everything is going to be the same one. Thick filament. What is a thick filament called? Thick one. Remember?
Myosin.
Okay. Thick one here. This one. Is it called like a myosin? And the thin one, the red color, which will denote that as an actin. So actin is a protein, and myosin is also a protein. But this arrangement, okay, it's when you see under the X-ray pattern, if you have electron microscope, and as well as if you do the X-ray pattern of this protein, how arrangement, I mean, there are three different bands which they used to see. Okay. When you see on this portion, see when you look under this by X-ray analysis of this particular area only, okay, up to this, call it as, it comes like this. This is the one region, and then the another region at the middle, okay, one region, and then if you see like this through only this particular area, then you will be able to find like this. There's the third region. I'll write a little bit of things here. Read it. So when you see only this area, okay, under the X-ray pattern, so it looks like it reflects over here, okay, it reflects like a star, like one star like this. Okay. You are looking using your X-ray crystallographic studies, so you see only this part. In a way, it forms like a pattern. Like this will connected, and this being connected, this being connected, this is being connected, this like that. It comes out. So what you are observing is here only the myosin, just the dark one. Okay. When you see only this part, okay, under the X-ray arrangement or X-ray crystallographic studies, you will find like a dot, a thin dot, and they have a very good pattern of arrangement, like, you know, it goes like this. So the dot, which is which in between that, you can see the thin dot. This is especially for, you can call it as an active actin band here. And then if you find some sort of a mixture of these two things over here. Here, if you look into through here, cut through here. So what you really see, here, just for your myosin, okay. And then what you get in between that, the myosin. This is something like a child play for putting in dots and everything, but it is a wonderful pattern which is being arranged. Okay. So if you see, the myosin is surrounded by the red actin dot. The myosin is the thick one and the actin is the thin one. So this type of pattern, which you will see that under the X-ray pattern or X-ray crystallographic patterns of the tissues. Okay. Now, the band, which we call it only if you look into that myosin alone, here to here, right? This they call it as a H band. H band. Otherwise, I put it over here. H band. H-B-A-N-D. H band. Okay. And then when compared to this middle part, we call it I band. I band. H-I band. Okay. And then if you see the junction of this actin, that they call it as a, they call it a Z. The Z line, they call it Z line. If you see in one pattern, they got Z line, which is the junction of this actin from one to the other, junction together. And then if you see only the, onto the myosin, which is going on to actin I band, myosin, yes. Myosin alone, they call it as an A band, myosin alone. Here, the myosin alone, they got the A band. A band. Okay. H band is a mixture of it comes out all combination of these and this together. It's some other proteins also been there. So it's not like a myosin alone, but it's myosin alone. It comes out like a A band. This one. This one. This one. So the H band is something like it's going along with this one. It's also, you call it as a H band, which is both together. But this pattern, if you see, you won't get this. You will get in a band something like this. Stars only. That's coming at the A band. That's for the A band. There's no in between that one. But here also you get, because of it goes one, the H and band like this. These are both similar together. But the A band is going to be the myosin alone. Okay. So H band is the combination of both.
So no actin.
No actin, no actin. In the A band, there's no actin. No. But H band, yes, both. You have got actin and myosin.
So which H band has no actin, or it does?
No, the H band is having actin.
Okay.
Actin. H band will actin, and A band no actin, and the I band no myosin. I band is only actin. You follow that? I'll write it. I'll write it here, like a H band. You get a myosin plus actin, and I band only actin, and then A band will have myosin. That's the summary you have to get. H, I, and A band. So you may have some questions on this. That's what I explained to you clearly. So when the actin myosin band, how far it works. It needs for the contraction. It needs what? Calcium ion. Okay, until the myosin band, and then ATP, which we have seen before. I'll explain this. When you use this muscle cell, you have mitochondria, nucleus, and the nucleus will give you the actin gene translator to the protein, and the myosin gene. Everything is being highly activated. When compared to the other protein, this actin and myosin dominates in the muscle cell. So the other proteins, which is not going to be expressed that much, maybe you have a maximum output of 90%, only actin and myosin. But rest of them are the sarcomere, sarcoplasm, like endoplasmic reticulum. You have got a different organelle. That's also, they do their own function. But the calcium and ATP, calcium is coming from the endoplasmic reticulum, and the ATP is from mitochondria. So they will help to function, to contract on these protein, that actin and myosin. As I mentioned before, the arrangement of this actin and myosin, in a sense, where is that here? I'll explain to you here. See, so they have got some sort of a junction with a tropo. I mean, there's you have a different join, so like it will go and sits over here, and thereby with the calcium, with the help of calcium ion and ATP, it will contract as well as it can relax in a position. That's what I explained to one of the animation. I'll go through the animation that will explain to you. So you have to remember, all this pattern is taking place inside the muscle cell. So don't imagine it is outside the cell, some fiber is contract and relax. No, don't think so. So please remember that each muscle cells, they have the actin and myosin. So if you have accumulation of these muscle cells, not one cell, but if you have more cells, okay, I'll write like this one, okay, another cell, muscle cell. So I'm just I'm arranging the muscle cells over here. Okay, muscle cells, all of the muscle cells now. So in these muscle cells, where do you find the nucleus? The nucleus will push in one side. Oh, here nucleus, and here the nucleus, here the nucleus, here the nucleus, and here the one other nucleus. So I'm pushing the nucleus as just I'm constructing a muscle tissue from muscle cell. Okay. Now you find the actin and myosin filaments, or you can call it as an individual protein. How they are arranged, if I see cross sections of these muscle cells, So I want to have the muscle cells, they connect to each other. This protein. These are red one, which I'm just getting, it's present all over the cells, but at the same time they can also connect with the junctions getting into. So now you get what? If this cell contract, that will reflect this one also. Do you follow? So the protein which is present in a way, the actin myosin filaments are the proteins which will contract, and at the same time all the cells, the neighboring cells attach to the neighboring cells, neighboring cells attach to the next one, and they put together, contract together. And then if they relax, and they relax together. Do you follow? I'm just going through from one single muscle cell, and I'm making the other cells, I mean how they are joining. See, these cells, just one direction, but if you see cross sections, and then these cells pile up the another cell, what happened, and these will go like a thread like thing, like that's the thread which is going on to neighboring cells and connecting next cells, and thereby it put together, it contract and relax. You follow now?
So then is the actin the one that?
Actin, myosin, both filaments. You need both of them, not only one. You need both. See, because the reason is if you have only one, like actin alone, like this, if it contracts, it cannot do that one. The problem is you have the myosin, which will have the arm. They call it as another arm like this. Okay, and this arm will go like this. It will bind together. So it works on a pair, So here this one, and then here it will go and bind. So it will push and then pull. So one act like a frame, the another one is moving on the sliding one to the other. Have you seen any sliding? If you have the slide, right? And the people, they go slide over there, right? So this is the one is pushing. This is the U. You are going on through the you have by gravitational as well as slanting position. So you can sliding over here. That's one aspect. But if you do in this respect, you can have it as an actin in one side. That's acting like that as a one frame. But the myosin, which is pushing over here, and then it will move. So in a different way, I explained to you that you need to have a calcium and you need to ATP. You need some force. ATP molecule is an energy and calcium. So it will help, like a one band, the another, and another, and another. So these are the one is actin, another myosin. So this arm will bind. If it is not binding, it won't move. So you need to bind with this arm to this tropomyosin to actin. So this will bind together, and this bind. So whenever this will contract, after the movement is over, then it will come down. So you will not get this contraction. So it's going to be a relaxed state about it. But when you want to do it, yes, you have to put together the arm, work together. So whenever there is a work, you have to get two fibers together, then it will work. And if you don't want work, it's going away. So for this, you need to have the calcium as well as the ATP, and that will drive the contractions and relaxation over here. Do you follow this part? Yes?
I have one question.
Yes, go ahead.
Proteins, so that means that they're inside and outside the cell connecting to other cells?
Yes, that's correct.
So they kind of like go across?
That's right.
So then, is with the C line would be when there's one cell and two cells, or no?
Which one?
Like the Z line, is that where?
Z line, which is present inside. Z line is the one which is the inside connecting the actin inside the cell. But at the same time, the Z line will also go through the next cell as well. Can pass through that one. Okay, and thereby you will see how the cell is being connected together.
So the C line is not the cell junction?
No, it's not a cell junction. No, it is not a cell junction. I'll tell you here. And the one, I think we have this picture over here. Here the myofibril, here the Z band, which is the one cell. This is a one single cell. And sarcoplasmic reticulum, like endoplasmic reticulum, you have these are the sarcoplasmic reticulum. You concentrate upon only the blue, the dark blue and light blue, and then the sideways, you can see the side view of that. This is a one single cell. Imagine the nucleus is pushing in one side, and then you have the mitochondria, and then how these cells are arranging in a fiber, So it goes through, this will go through the next cell. This will go through the next cells. The Z line is present inside the single cell. So the another cells also. But it acts together, all cells, because joined together so they can act together. Suppose if you are lacking calcium, if you don't have ATP, then you have got tired. You cannot do, muscles become weak. and also you have this nucleus and more of glycolysis which is taking place in that muscle cell, and that will produce more of lactic acids. So another thing, if you do overworked the muscle, if you do too much of work, and that will get into your nerve endings, and then you get some pain, and then you have to do some relaxation. You cannot do exercise all on a sudden. I think you have baseball players and softball players here in JAG team here. So they cannot just like that go and take a thing and to play. They have some warm up exercise, right? You do that one? Come on, Victoria, Brock Heyman or Kayla. Are you there? No?
Yes.
What is the purpose of doing warm up exercise before you play a game?
Warm up your muscles.
Huh? Warming up?
Warm up your muscles.
I couldn't hear you. Put the mic on.
So warm up your muscles.
Okay.
Pull it.
Toning the muscles. Okay. Not only one, but you do several exercise, right? Before you do that, right? Correct? Hema, what do you do normally? You are a pitcher or batter?
Yes.
Okay. What do you do before that?
As far as stretching,
Stretching. Okay. What muscles you are stretching?
hamstrings, quads,
Okay.
the muscles in your arm.
Okay. If you are not doing the stretching, what will happen?
Well, everything's going to be tight.
Ah, there you go. See? So he is giving a training to his muscle first before he used to play. So he is giving his, is a game, but he has to train first so that whenever he wants response, and the muscle should activate quickly. Otherwise, what happens? It won't. Correct? So basically, what you are doing it here, which is mainly on this myofibril and Z band, it should be in a good shape, in a functionable aspect on that. So you are making sure all the system is working very well, and then you are going to play. I mean, that's one of the reason that we need to, to get the mechanism, how the mechanism of individual muscle cell will reflect onto the whole tissue. Okay, when you see this muscle, which is muscle cell, which is translated into a different region of muscles, I mean different types of muscle, like a smooth muscles, as well as in cardiac muscles. Okay, let us see here in one of the picture here. Okay, here sliding filaments. Just I'll show you here a little bit of that Z line and how they are arranging it. Okay, myromycin and heavy chain. I mean, these are relaxed state and contractile state. I think I have given this particular picture into your book, into your blackboard as such, so you can check together. Then how these cells have been arranged here. You can see this one here, the bundles, all of them put together. Okay, so you get the dark and light bands now. What is the dark and light band, which already you have seen in your muscle several times? So here the Z line, which is coming into the actin, which is a thin, and then you have a myosin and actin together, and that you have a thick one. So that's what we call it as a striation when you see onto the muscle tissue. So if I ask a question, short notes on striation, what you have to do, you have to explain the striation is the result of the protein or muscle protein, especially actin and myosin. When you see the actin, that's a very light band. When you have the actin and myosin sliding together because of the two protein together, so you have on the thick band, and that is coming as together. That's a dark and light dark. So alternatively, you get a dark and light band. Okay. And if you see under the next cells, I mean one cell into the other cell, which I mentioned earlier. Could you see this picture? I'll just explain to you. This one. Okay, this is the intercalated disc or the longitudinal sections. Okay, these are the cell junctions. How the cell junctions, how they arranged here, intercalated disc over here. Could you see this projection of one cell and the projection of the other cell, next cell? They put together the joint. So this is the region where the cell junctions is there. So that's what the Lillian asked the question. The Z lines are over there. Z lines present everywhere, but at the same time, the connection of one muscle cell to the other cell, you find the actin and myosin filament, they are joining together. So this is the junction, cell junction. That's why you get a whole stretch. Though this part is one cell, this is another cell, or this is another cell, just it goes on, if you see in a broader picture, you get these striations. If you look as if it is something like a membrane, but it is not like a membrane. It is made up of different cells, but individually, each cells will have actin, myosin, that is joining together and that form the pattern like a striations. If you see in the cardiac muscle cell, if you see this one, intercalated disc is the one other thing here. I'll explain to you here. Okay, cardiac muscle intercalated discs over. You can see that the arrow which is over here and this is one of the electron microscopy of the intercalated discs. If you see this arrow, it's very, you cannot see clearly, but when you see under the microscope, even in the light microscope, you will find a thin line-like thing. I think in our lab, we discussed about our slideshow. If you see cardiac muscle, you find a thin line in between the dark and light band. That's the one which is the intercalated discs, which is coming out over there. Just cell junctions, collection. Maybe the intercalated disc you will see, in between the cell junctions. And here you can see the sarcoplasmic reticulum on the cardiac muscle over this one, and this one. It's a sarcoplasmic reticulum. Like endoplasmic reticulum in a normal cell, but it's the muscle cells, sarcoplasmic reticulum. Okay. And I'll just go through some of the other cells here, how the muscle cells will look like in a smooth muscle cell. See, this is a one smooth muscle cell. Okay, one smooth, like a sphere shape, one smooth muscle cell. But if you see the cross section of that, so you have individual cells of different cells. If you cross it, this is a one single cell. Okay. So the smooth muscle cells, if you see this fiber, you can see this one here, this fiber, one cell, but this is a tapering end, and also the nucleus is there. But here the nucleus is present at the center, center of the cell. But at the same time, you find the thick and the thin actin filaments here, thin actin and myosin filament in single cell. Okay. So if you see on the skeletal muscle, the regeneration, how the skeletal muscles are regenerate in one of the light microscopic pictures over here. You see a skeletal muscle. Here you can see the striations. This is a tiny one. I don't know whether you can see it clearly. Yes. And this is the degenerator of the upper is the normal one. As the degenerator, this is a degenerator of this muscle tissue. And then after a while, it has been regeneration of the reparative process, regeneration here of the muscle. Again, it's going to be regenerated. So these are the histology of how the muscle is being damaged and then is coming back onto the normal of the regeneration of that. So when you do a severe exercise about this, what normally you get the muscle become muscle cell, which I mentioned before. Okay, so this is you have the nucleus over here. Okay, and these are the actin and myosin. Okay. So what you do when you do a severe exercise of this, so this muscle cell, I just draw in a different way, it's easy to remember. This is the one, this is the muscle cell, this is the nucleus and this actin myosin. Then after you have doing a severe workout, so these muscle cells will go like this. So what you find here, the size of this one is increased. Size, it increased. This is small. Before, I'll do that. I'm going wide. Okay, now. Okay, good. This is small and this is a large size. So this is the before. This is the after of what? The exercise. So before you thin muscles, and you get after exercise you get a strong muscles. Do you think the muscle number increase here? No. That's what I showed you here. The people, they misunderstood. They got, hey, you have a muscle, more muscles will develop when you do the exercise. It is not like a muscle as such. But you are increasing what? The size, you are increasing it from smaller size to larger size. At the same time, you are increasing the actin, myosin, protein. You imagine. Here you maybe have a 25, but here you have a 225. So how much you are going to gain, you know? So that gives as a muscle, as a toning, as well as, you know, the muscle mass, they call it. Mass, they call it. The mass is not the number of muscle cell, but the size. So we call this aspect of this one called hypertrophy. Hypertrophy. That is increase in size. You follow? The muscle, if you do the exercise, you are giving stress, more stress, and you are doing regular exercise, and then you're getting hypertrophy. Doing good exercise, get it. But what the people, they used to take some physiological thing like a steroid. What is the work of the steroid? In the muscle, you get steroid injections and then makes them more of the muscular body. Why? And that is being that's been allowed in a sportsman, but not like a display artist. That's a different story. You the muscle man, but normally you the players and game, they discourage. They don't expect any steroidal supplements or steroidal activating supplement or anything else. Why? What the steroids will do? Feeling good?
Boost your energy level.
Boost your energy level?
Yeah, so you can relax more.
Huh?
You can exercise more.
Exercise more. Which one? Do you think that it increases your actin or myosin?
No, it's not.
Then for what? Why do you?
Is it just hormones, Dr. Soma?
Yeah, it is a hormone, right? That's fine. But what is the function of the hormone in the muscle? Well, why the sportsmen and they used to inject R, and then you want to exceed the competitors? Why do they use it? Steroids.
Stimulate.
One of the function of the steroids, they act on genomic level. They act directly onto the DNA, and not only they expect your main aim is to produce more of actin and myosin. That's why I'm coming to the point where we started. Yeah. So if you have more actin, myosin, then you have a more energy you feel, because you can contract and relax your muscles. Your muscle can obey you, and then you can do whatever you want to do. And also the action. Another one is the metabolism, which is being be elevated. That's what one of the things you thought. But you have a lot of side effects, because you are unnecessarily damaging your genome. In a sense, you are activating the gene. Yes, it will increase your muscle mass, no doubt about it. But at the same time, if you are not doing the exercise, in the end, the steroids will lead to obesity. It will lead to more of accumulation of fat, and then the third part is for the long-term use of steroid in their lifetime, maybe 5 to 10 years if they start using it. then at the age of 70 or 60 years, these person definitely 100% they will get cancer. No doubt about it. Steroids. So the doctors even now they are teaching the steroid therapy for some arthritic patients, some of them. But that is good for a while, for a temporary. But if patients live long, more than 70, 80 years, definitely they will go under cancer because of the steroid, not from anything else. And this is another thing which we discussed earlier in our on the cancer Cafe Scientific. You have the gun, but who is going to trigger the gun, right? So some of the environmental factors, some of the dietary factors, some of the habits, those are the triggering mechanism which will induce the cancer. One among them is the steroids here. You won't see immediately, but at the later part of the life they'll do. The doctor, who cares? After 70 years old, everybody's going to die. Why do you care about it? Give steroid. Be happy. So that's what they used to do. So that's the story. But now coming to our point in the steroid, the role of steroids in sports. So whenever you study on muscles, and you have to always think on this part, how the steroids will activate the muscles. Especially the answer is the steroids activate the gene expression of actin and myosin, so that will increase the number. Okay. What I'll do, I'll give you a break for 10 minutes, and then we will be back, and then I'll go through some of them, and then we will review for your exam and next week, and to discuss about the short answer questions as well in the class. Okay? See you in a bit. Any latecomers?
Shazia.
Shazia. Okay.
Amanda Martinez.
Aha. Amanda. In Victoria. Amanda Martinez.
Yes.
Okay. Anybody else from Victoria? Nope. Okay. Shashia Khan. Okay, that's what I got it. Okay, now. Okay, this is the PowerPoint which already I've given to you in your Blackboard. So, and this is a presentation too. Given the objectives, why we study in muscle tissues, what we study here, what is the objective. We want to study structure of skeletal muscle fascicles, fibers, myofibers, myofilaments, and that's a thing which we discussed earlier now about the actin and myosin, and put together they form the fibrils and filaments and everything. We will see now about the Purkinje fibers, that's in cardiac muscle tissue. Okay. Sliding filament or concept of contraction, so that's what we discussed earlier. And we will see if it moves, it is alive. If it is not moving, it is dead. Right? So hydra to human muscle tissue declared the life. So if there is no life, what? If you see a dead person is moving, then he is alive, right? Is it? What? Ghost? Zombies? What is zombie? Are you believing in zombies? Anybody is believing in zombies? They're in Halloween. Okay. Just the stories for children. Okay, the rhythmic contraction of the heart or sluggish peristaltic action of intestine. That's how the smooth muscle cell contractions, right? And cardiac muscles of our heart. And the contractility is inherent nature of muscle, but conductivity is the inherent nature of the nerves. So we will see the conductivity next class. I mean, after the of the exam, we will see that one, nerves, tissues. Okay?
You said after the exam? I'm sorry. I didn't
After
see-
the exam.
Okay.
We'll see that after the exam. Okay. Now, we'll go on to connective tissue and muscle tissue. Connective tissue and muscle tissue together. Functional role, both of them, they have the contraction. And the connective tissue is the way to force the contraction, direct the force of the contraction. Okay, function of connective tissue. Connective tissue provide passageway for capillaries and nerves that supply the muscle cell. So you have to understand like an organ system here. We are talking about two tissues. Connective tissue helps the muscle tissue. In the sense, connective tissue is a liquid connective tissue as a blood and that will and the nervous system, everything put together into in the muscular tissues. I explained one little bit of information about the suppose if this is muscles which is going over there, you have a dog bands and everything, and onto the top you get here this neuron which is coming over here, and then the neuron is end, the axons ends over here, and this is a muscle, M for muscle, and this is for the I just put this the neuron, N for neuron, and this one is the junction of neuromuscular. NM is the neuromuscular junction. Imagine what type of architecture here in this neuromuscular junction. So nerve ending, and then there's an axon and these muscle cells, and there is a separate. Sorry? Anybody? Oh. Okay, if this junction will increase the output from the neuron, that's suppose if you think I need to lift my arm, what actions is going to, is taking place in our body. If you think your brain and the brain through the neurons, and the neurons will go the command to the muscle cell or muscular junction, and here it gives the order to this. So neurotransmitters, and this will activate the muscle to contract. So it's something like a brain and then activate the neurons and to the muscle. So this architecture, the neuromuscular junction, is a specialized one. We will discuss later on when we discuss about the nervous tissue. It's a very interesting one. So neuromuscular junctions, and then how the neurons conduct the message, how the muscles learn the language, or how it will understand the order which is coming from here. It's interesting part because I'm dealing with the muscle. I will touch up with this and before I go into the nervous tissues the next week, not the next week, the following week. Okay. Go back. Type of muscle fibers which we did see now: smooth muscle fibers, and then predominant tissue involves of blood vessels. So the blood vessels is and ducts and hollow viscera, all are smooth muscle cell, right? Sheet-like layer. So it's like a sheet-like layer. Cardiac fiber found only in the heart and walls of large artery vessels, the cardiac fibers. Skeletal muscles, as you know about all the skeleton muscle attached to the bone. Okay. Let's go on development in vertebrates. That's on muscle development in mesoderm. This is for a developmental aspect from muscles coming from mesoderm, except the intrinsic muscle. So you have some questions from your, I think from multiple choice questions over here. And then mammalian embryo, 2 stages of muscle development: histogenesis, another one is morphogenesis. Okay, just read through that. And you have again the skeletal muscles from mesodermal somites and cardiac muscles from primitive heart fibers. So they are coming from this splanchnic mesoderm. Splanchnic is the abdomen. This is what is splanchnic, where the intestinal blood supply, we call it as a splanchnic circulation, splanchnic mesoderm. So that was splanchnic. You know, the network of the arteries which is present in the intestine. That's we call it splanchnic. But the cardiac muscle fibers is developed from the splanchnic mesoderm. That's on the developmental aspects. Then once it's been developed from that, then this is being separate. Cardiac muscle fibers separate from the intestinal tissue or muscle tissues. Okay, smooth muscles, the most primitive of the three forms is the muscles arise from the splanchnic mesoderm. Smooth muscles also from mesoderm. Okay, splanchnic mesoderm. These are the developmental aspects of where the muscle tissue is coming from, origin. And then this we have already seen some of the light microscopy of muscle and size of muscle. You can see the individual 20 to 200 micrometer depending on the organs. You can see, and especially on pregnant uterus, the length of the smooth muscle that it increased 600 micrometer. Imagine. So that is again the cell size is being increased. We discussed earlier is hypertrophy. Okay, this is also something like hypertrophy because you are giving more weight and weight is increasing day by day. So you have the increasing the size of the muscle tissue. So the diameter may vary 3 to 9 micrometer. These are the size of smooth muscle. Okay. Component of smooth muscle, which we have already seen some of the cigar-shaped nucleus and cytoplasm we call sarcoplasm, and contractile elements are myofilaments. That is nothing but myofilaments is nothing but actin and myosin protein filaments are the delicate cell membrane that cannot be seen by light microscope. We cannot see that one in a LUM or a light microscope. And these component of smooth muscle cells, you have innervated by autonomic nervous system. As is mentioned before, smooth muscle is not voluntary. Voluntary muscles are skeletal muscle. They are involuntary, meaning you don't have a control of your own heart contraction or the lung contraction or your stomach contraction, except you can put more of oxygen or your air, you breathe in, breathe out. That's what you can do it. Okay, that's a part of your skeletal muscle as well. But in the smooth muscle cells, as such, in inside your body organs, yes, you don't have a control over it. So that's why we have to be very careful. And not every fiber receives a nerve terminal. Again, all these internal organ or the autonomic nervous system present in the smooth muscle cell. And that is it's on its own. It's not be sensitive what you want to do it. No, you cannot do anything on that part. The stimulus passes from one fiber to another by gap junction. That's called nexus. And here this is not O. This is a typo. Capillaries core in connective tissue that inverse the muscle bundles, not individual fibers. See, this another one that I want to highlight here. When you see the muscle bundles, right? If you have this is going like this in the connective tissues, which is connecting it. Suppose this is one cell, this is another cell, but they are connecting together, right? This is another cell. But these fibers, as I mentioned, they are running through. But if you see these bundles, they are not giving the individual cells is not supplied with the blood supply. Instead, the whole thing is being covered, the whole bundle, because the bundle is mean several filaments which is going to be, right? So this one is something like I draw this dark one or the black one which is going over here, and this is the blood supply. Blood supply over here. This is connective tissue, but inside that you get this one. So this is a cross section. Okay. So individual actin filament, the fiber which is running through inside. This red one and actin myosin. So it is not supplied to individual thing, but it gives a collection of the bundles. So all of the bundle is being surrounded by the connective tissue where you get the blood supply. So that's what I want to mention here. Okay. Skeletal muscle, voluntary forms, and this already we discussed. All striated muscles, but not except cardiac muscle, right? Cardiac muscle also is striation, but that's having different characteristic. But generally, the striations is one of the diagnostic feature for the skeletal muscle. Okay, component again. Here we have seen many of those details of earlier classes. And shape of skeletal muscle, giant multinucleated, cylindrical, and we have discussed earlier now, even now we did with the dark and light bands and everything. Fibrils, dark and light bands of one fibril are accurately aligned and registered with the dark and light bands. So that's what we discussed today. And dark band is called anisotropic or A band or doubly refractive. These are the X-ray diffraction pattern, which I just explained to you. So you may get a question from these slides too.
Which one was that?
The A band, B band, A band, H band, and Z band.
Okay.
Red and white muscles. I don't know whether the red meat and white meat is the same or different. When you go and see, you should eat only white meat or you should eat both white and red meat. Is that the same meat is something like a muscle if you think you translate into that. So red and white muscles. So in this context, in fresh and skeletal muscle are all distinguished as red and white fibers. So they have the muscle fibers, the red and white fibers. Red color is due to abundance of muscle pigments, myoglobin, less glycogen, but an increase in mitochondria and richer in vascularity. So red color that denotes more of the myoglobin. Okay. And also you have a pale, large white fibers, fewer mitochondria, less pigment, and a poor blood supply. So they have the different characteristics. So you have the skeletal muscle, you have red and white fibers, but they for this red one you have a more mitochondria, meaning you have a hyperactivity. You have more contraction. So if this muscle is contraction more and it gives a more energy, is coming over here because of mitochondria. So the pale, large white fibers, they have fewer mitochondria, less pigment, the poor blood supply. So it may give a supportive thing, but also do a contraction, but it is a less when compared to red color or muscle fibers or red fibers. Let us see what those things are. Functionally, red and white muscles are different. Red muscle contract more slowly than white muscle and called slow muscle and do not fatigue easily. Okay. And white muscle contract fast, example leg muscles, and used in running and fatigue quickly. Which is correct? Red or white?
Sorry, repeat that.
If you want to run fast, which muscle be activated here? Which muscle contract
Red.
fast? Red? White.
White?
Yeah,
I'm sorry, what?
that's what I'm saying.
Oh, okay.
It looks like when I previous slide which I showed you, you more of mitochondria, you expect that more action, but it is not.
Oh, it's
that
not.
way. That's what I'm
Okay.
saying. Contract more slowly than the white muscle and called slow muscle. They call it a slow muscle. Red muscles are slow muscle. Okay. White muscles are contract fast. But at the same time, they fatigue quickly, white muscles, because of the hyperactivity. Muscle of leg. Now, we are going on the muscle of leg. If you are running, how many of your marathon runners here? No one? Running to the school, that itself is a marathon, is it? Okay. Because I know Victoria is they have sportsmen over there, but here we don't have a sports team, anything here. But we will start soon here in this location. Okay. So we have the gastrocnemius muscle, white fiber duration of contraction providing velocity fast 1 per 30 seconds. Gastrocnemius muscle. That's the fast. Okay. Another one is the adjacent soleus muscle with the red fibers that has a contraction of 1 in 10. So you look at that, fast and slow. Red is acting slowly. So the gastrocnemius can contract rapidly and forcibly, providing great velocity for leaping and running. So this is the one which we need. We have to develop of more of gastrocnemius. That gives more of energy. Soleus is a postural muscle involved in giving support against the gravity. That's what it will do. Soleus muscle or red fibers gives only the support. So if you run and then practice more, and you are exercising this gastrocnemius. Okay, that's what we need to do. neuromuscular spindles, so the another one which I explained before the class. I'll come back to you when we teach in the nervous system. Again, exhibit slow and fast muscle and long and thick bag of fibers and slow muscle that act in a tonic fashion. That's a neuromuscular spindle. This is another type of muscle fibers over. Okay, now we've got cardiac muscle, bulk of walls and septa of the heart origin in great vessels. They have striations, and also you have the myofibrils, nucleus around very prominent nuclei. That's what we see under the microscope in our lab. Okay. Muscle fiber, this is a cardiac muscle fiber, has a thin sarcolemma and over prominent nucleus, and comparatively shorter than the other fibers in branch form complicated network. This is a bit complicated network, but these are the light microscopic picture of dark irregular lines. And the intercalated disc is a diagnostic feature of the skeletal muscle to cardiac muscle. So cardiac muscle will have the intercalated disc also branching, whereas the skeletal muscle, they have the striation but not the intercalated discs. Okay. So it will help the passing across the cardiac fibers, signals to contract the muscle tissue. The electron microscopy, I put EM, that reveals the intercalated disc to be adhesive connecting junctions between separate cells, and they have the excess of blood capillaries, cardiac muscle. They need more of oxygen and nutrients to cardiac cells to sustain the vigorous, continuous heartbeat. More mitochondria for high demand of energy. So we did, comprehensive of all the muscle cells so far. I want to give one simple, our own experiments when I was in a graduate school. When you do the cardiac muscle, just now I explained to you, is a vigorous and continuous heartbeat. Suppose if this heart, it's coming out here, and then you have it's going, the aorta just going over this is a car. So this, if you make a clamp onto this artery, a little away from this aorta, so what will happen, this heart? I'm just blocking by a clamp. So what this heart will do? Yes? Suppose if you send a hose tube for getting a water in your lawn, and then all of a sudden you have a block in somewhere, what will happen to that? Not block, but you are...
There will be more blood in the heart?
Yes, but what happened, the force, you have a increased force, right? So which part, which is because this is coming from ventricle, it is going out to the throughout the body. So when you make a constriction in the aorta where the blood is going out from the heart, when you do the constriction over there, the ventricles become more stressed by contracting more heavily because it has to pump hard because of the constriction here. So what will happen here? So this cardiac muscle on the left of the ventricle become hypertrophy. Hypertrophy. Okay? Is being activated. So another aspect is, this is one of the model which we do for the hypertension, hypertrophy and hypertension, or the increasing the blood pressure. And all this is a model which I'm explaining to you. So if you constrict here, constriction, and then that will induces a more of the force. So if naturally how it occurs here, you got the cholesterol or plaque, or if you have some sort of atherosclerosis in this condition, you get excess of the blockage of the arteries that in turn translate into the excessive active heart muscle, especially on the ventricle, and that become the heart become hyperactive, and then hypertension is the result on that part too. Okay, now go back now. Here, the vigorous heartbeat and more mitochondria for high demand of energy. So that is the part that will lead to hypertension. Purkinje fibers, that's on the modified cardiac muscle, is also a separate one, because I asked a short notes on Purkinje fibers. That's another question. So I expect all this part of the Purkinje fibers, which is specialized for the conduction of electrical impulse and similar to those nerves, neurons. Okay, it goes like this, like in the heart, and you have the fibers, which is going to the one side and to the other. So these are the fibers, just going over there. But it's not like a muscle, but looks like a muscle tissue, but it's a modified one. And these fibers will activate, just like a neurons, it will activate the muscle to contract. As I mentioned earlier, like a neuromuscular junction. And these fibers, which are specialized cardiac muscle fibers, and they will activate the muscle, cardiac muscle. That's why you call it as a Purkinje fibers, and it acts through the septum as the bundle of His. This is a specialized and modified protein of these muscle fibers. They found in the Purkinje fibers are found in sinoatrial or atrioventricular nodes. So you have to mention what is Purkinje fibers and where do you find this one. Okay, in interverticular septum as well as the sinoatrial and atrioventricular nodes, and they are thicker and large fiber than cardiac and contain the much in glycogen. So Purkinje fibers contains glycogen for its own energy resource for that. So it helps in the maintaining the cardiac function. That's very important. Blood supply to the heart, coronary artery. That's what normally used to have a very thin artery, coronary artery. But you have to have a maximum blood supply. So that's why if there is a blood supply to your ventricle is blocked, you have to have an angioplast, right? So you have to have a new blood vessels. So you have to surgically. You have to attach another artery to supply the blood to the heart. So extensive blood and lymph capillaries and surround each cardiac and Purkinje fiber. So you have to have more blood supply as well as the nerve supply, autonomic nervous system again, and nerve fibers do not generate the heartbeat, but the individual fibers cells that will activate. That's one thing. The nerve fibers do not generate heartbeat, but the nerve cells in the sinoatrial node do generate the heartbeat. So you have to remember what create the heartbeat. The nerve cells in the sinoatrial node. That's what I explained earlier, the neuromuscular junction that make a big difference here. Okay, here the nerve cells that in sinoatrial node to generate heartbeat. So once the heartbeat is being generated, then that is being communicated to the next. All the heart cells will have inherent quality, contract and relax. But something it should be stimulation that's from the nerve cell, from autonomic nervous system. Okay. They do affect the heart rhythm. If you stop this, the rhythm of the heart. So that's why I don't know whether I can go detail on this part, but we will study during physiology class like a P wave, Q wave, or PQRS, T wave like this. Have you studied in your physiology class? Because we had a comparative physiology class when we discussed about the PQRS, something like just going over here. Like that? Have you studied? Right? So when you use the ECG, you can see like this, right? So here you got the PQRST, PQRST is going over there. So we did study extensively of these peaks and trough and everything, and that is mainly of the contraction and of these heart muscles, when it goes to the blood is going from atrium to the ventricles and ventricle to the right and left, and all those things we can study in a graphical way. That's on the physiology. I'm not going to take that class now, but just remember that they affect this nerve fiber, they affect the heart rhythm, the pattern. Okay. Sympathetic activation speeds up the heartbeat, and stimulation of parasympathetic nerve slows the heart. So you have to have two types. Parasympathetic nerve slows the heartbeat, and sympathetic activation that speeds up the heartbeat. So these two we will study in next class about when we discuss about the nervous system. Okay. So whenever you want to make, sometimes you have an exam fear or something like, and then you have a heartbeat is going up because of a fear. So that gives the activation of sympathetic activation. And sometimes you have to have very calm and quiet and do some sort of go before or after quiet sleep or good food or something, listening to a music or something, good music, not like the speed of the heart, but normal music that will quiet the heartbeat. So you have to we will study when we discuss about the nervous system too. Okay, now we will go any questions so far this class? Any questions? No questions? Is it good news or bad news for me? Any questions from Victoria? Cinco Ranch? No questions? Okay, that's good. We'll go on the exam now. We have to review for this exam, right? Exam 2. You will get the multiple choice questions and short answer questions. Essay for short answer questions. This you have around 130 questions. This one. This you will finish up maybe 15-20 minutes, right? Multiple choice question. Each questions probably half a second or 2 seconds. Half a minute. Sorry, half a minute. Okay, you had to read. Okay, I agree that you had to read, answer, fine. So I'll give you probably half a minute is going to be 65 for all this answering the question. Half a minute for each question. But you are going to A, B, you are going to click, right? How much time you are taking for clicking? You are not writing it, you are reading it. So improve your reading skill, and then improve your clicking skill. Clicking is, it's very simple, right? So if you do click, it's very simple. Okay, so you have 2 hours time, and you are going to answer the short answer questions here, around 10 questions. Okay. You have to write something. That's what is lacking last time. They are writing a word, one word, and then even that one word is being condensed into an abbreviation. If you say yes or no, you said why. I don't know what the why means. You say yes. So don't do that.
So you want sentences.
I want the short answer questions in sentence. You have to write. You have to write a noun, a verb, pronoun, adjective, okay, and then a period. Okay. And then you start another sentence with the same thing, and then another period, another period, another period. So like that, you have to write at least 5 period, 5 periods, at least 5 periods. Yeah.
For each answer?
For each question,
For each question, I mean?
yes.
You want 5 sentences for each
With
question?
periods, yes. Sometimes
Okay.
people they write. I mean, I'll tell you. I did the muscle fibers. They got the fib.
Period.
Okay. Striation.
Period.
Okay. Muscle fibers. There is no verb. Muscle fibers striation. What do you mean? If you write like that. So I'm asking the question of skeletal muscle. If you say muscle, you have to write the muscle fiber. You can start the answer from the question itself. The muscle, the skeletal muscle fibers contain striation, or is made up of actin and myosin, and this the different bands and everything. So please try to write. But I'm not saying that you don't know how to write of the sentence. I agree. You are 100% perfect English, more than me. You are speaking, you are writing, good, fine. But why don't you write in the exam? That's my point. That's why I want to tell you about it and how to write. So if you don't, then I have to ask you to write down by dictate question and answer. I will dictate a question and then you have to take down the answer, and then I expect what I said you have to get onto the paper, word by word.
How many short answer questions do we expect?
10? You said 10.
10. What are those 10? Okay. Did you attend your class, regular class? Okay, I mentioned every class. These are the short answer questions. Take down that questions. Okay. So to start with, we will go through this muscle tissue today. What short questions you will get. Number 1. Okay.
So are these worth, the 10 questions, are they worth like 50% of the?
That is correct. The value, I didn't give it to the value. Okay. 160 questions, 135 or 30 short multiple choice question will go around 60%. Okay. And then 40% is going for the short answer question. Short answer questions for 40%. So if you have done all right, all 130 questions for you are getting D, definitely. No problem. Absolutely. But if you are not writing it short answer, but you are writing only MCQ, you are sure you are getting D, provided everything is right. Okay. If you do a short answer questions, then you have add everything put together. If you write 2 questions and then you are leaving out the 8 of the other questions, 10 times 4, it won't be 40. Okay. So these 4 point will be divided into your at least 4 bullet point or your sentence. 4 sentence at least you should write it, each sentence. You have to read all those. Okay, what are the questions now? Coming back. Okay. It's a general. Another clue I can give you. Some of the questions you get an answer from your multiple choice questions. You see the MCQ, you get some answers. So don't write that same thing, but you have to make a sentence. That's what I want. Okay. So in the muscle tissue, you see that one here clearly? Okay. Morphology. Okay. Function. Morphology, function of skeletal muscle, smooth muscle, cardiac muscle. Morphology means how it looks like. What is it? And then you are going to write function. So this is the question number 1. Not question number 1, but you will get the content from this. You get a question, not the exactly the same wording, but you will get the morphology and function. But I'm right, like a describe morphology of skeletal muscle, or I'll ask you to describe the function of the smooth muscle cells and then cardiac muscle cells. Okay. Also, you can include morphology, function, and where do you find. I put it and location. Location is very important. Location, location, location. Anybody's realtors here? Okay. Question 1. 2. Okay. So you have to write all these contractility, about these functions and movements, right? But 2, you have to study what are the actin and myosin. Separate question. Okay, actin and myosin, and I expect you to write about the different bands. H band, I band, Z band, and A band. All those things out. You have to write it in a sentence form. Actin and myosin bands. Right. Okay. Number 2. Number 3. Short notes on, you have to write about the intercalated disc. Intercalated discs. If I say short notes, intercalated discs present in cardiac muscles. Don't say that, but you have to include it is present there, and you have to write. But this is the question. But what I expect from you, location, where do you find, and then what is the function, how they are arranged. Okay, so all these details, then you have to write it up. Intercalated discs. Okay, so this is the answer. 4. Write short notes on connective tissue. We discussed just now. Connective tissue and muscle tissue. For this, I expect the answer, like a connective tissue here is a blood vessel, how the blood vessels is formed in the actin and myosin, and then it supplies the blood to the muscle tissue, and it supplies not individual muscle fibers, but is a bundles the blood supply, and also you have a collagen and also adipose tissue, also a connective tissue. So where do you find, and all those details you are going to write it here. Okay, that's number 4, connective tissue, and then for 5, and then short notes on nerve tissue and the muscle tissue. What is the role of nerve tissues and muscle tissue? That's on muscle tissue. So you have to, I expect from you, how the neurons or the neuromuscular junctions, Here you can read through some of the aspects of neuromuscular junctions, where the signals. We are going to discuss in the next class as well, but just I want to say to you where the neuromuscular junctions you have to write. 5. Okay, now I shall go on to the my next the previous lecture. Yes, I'm giving your questions from the PowerPoints, so that is easy for you to write and study. Okay. What we did in the last class and function, sorry.
We're having that question over the lineage of blood cells, right?
Yes. Did I give that question last time?
You said last time.
Yes, that's right. I'll go through that one. Yeah. What else we discussed earlier? That's on the circulatory system, right? Okay. So and the connective tissue components of muscle tissues. One of the thing I expect from you, one of the PowerPoint which we did with the endomysium, perimysium, epimysium, and those things also you are going to include onto the answer. I expect the connective tissues and muscle tissue, where do you find the the endo, epimysium, and perimysium, endomysium. So you have to study about where do you find endomysium, epimysium, perimysium, and all those things also, which I gave in the previous lecture, and the PowerPoint is there answer. So you have to write those. That's the same question, but I mean I gave the answer. Now what I expect from you, okay? So that's another thing, and we'll go on to our lecture 5. Let's go on to lecture 5. We are going to cover 4, 5, 6, 7. So that's for this exam, correct? Okay, in the circulatory system 3, that's on fluid connective tissue. And the question question number 6, which we did with the blood cell origin, correct? We did discuss that one?
Yes.
And different type of cells, and which is we do the hemocytoblast, and then it goes on different one, right? So you have so those you have to study. Good question on that part. Okay, you may get 2 questions from that blood cell origin, 6 and 7 also blood cells origin. So different questions you may be asked from that. So 7 questions over now. And then we also found the difference between serum and plasma. That one. Okay. And 9. Short notes on, you have either RBC, red blood cell, okay, or you have to study about the white blood cells. So either one you will be asked, either this or that. Okay. Here I expect where the origin of RBC, how the RBC is being produced. So one is coming from here, but the function, the number of RBC present in the blood, and then the function, and what is the structure or the morphology, and then where it will die. We did study about the spleen and everything, where the fate means the origin and the metabolism, and then the death of RBC, fate of RBC. And then the WBC or white blood cell, I expect the answer mainly from the different I'll write it in separate. What I expect. White blood cell, I expect from you what are the white blood cells which is available, like a polymorphonucleus, neutrophils, lymphocytes, monocytes, macrophage, right? So here you are going to write the origin again. We have covered the earlier question, but I need all of them for your function from here. Okay, so that's why it's in question number 9, and then question number 10, because I'll give you a few questions more so that you have to prepare. That's been covered, basophil, B lymphocytes, and every cell mediator, everything. So go for lecture 4. Got the connective tissue. We have got a couple of questions over there. Cartilage and bones, right? Which we studied earlier. So, distinctive properties of cartilage and bones. The properties of cartilages and bones. Here I expect from you what the characteristics of the cartilages. Don't say cartilages are hyaline cartilage and then fibrocartilage, and not like that, but you have to give what is the major difference between cartilage and on the bone. Example, in matrix, what is the difference? And then vascular or blood supply, vascularity or blood supply. Okay, and then the different types of functional variability. So these are some of the answer which I expect from you from the properties of the cartilage and bones. Okay, and then I need the hyaline, elastic cartilage, fibrocartilage, so that's everything that you have to study, and bone, osteon, and that's another one, 11, bone morphology, onto the morphology. So you have just a short notes about the osteon, Harvestian tubules, and on the neurons and nerve supply and everything onto that bone morphology. So these are all the questions for the short answer questions.